Good morning. Welcome to the Wigner Lecture. Just to remind everyone, the Eugene P. Wigner Distinguished Lecture Series in Science, Technology and Policy was launched in November 2013 in commemoration of ORNL's 70th anniversary. As all of you know, we just celebrated the 75th anniversary of the laboratory this past year. The Wigner Lecture is organized by our corporate fellows. The goal of this lecture series is to invigorate scientific discovery, spur technological innovation, initiate productive scientific policy debate. And we were just chatting with Sir Richard Roberts, just as all of you are coming in, and I couldn't help notice that there are a lot of early career scientists amongst us. And so I hope that these lecture series will also, and particularly people like Richard, will serve as an inspiration for all of you as you chart your scientific career here at this laboratory. The lectures honor Eugene Wigner, ORNL's patron saint. He is a chemical engineer by training, physicist by inclination. So those of you who are engineers like me, physics is still within our reach. Is the founder of the field of nuclear engineering, role model for coupling of basic and applied work. He's a research director of ORNL from 1946 to 1947 and winner of the Nobel Prize in Physics in 1963. Today, it is my pleasure to introduce you to the Wigner lecturer, Sir Richard Roberts, who is also a Nobel Prize winner. Richard shared the 1993 Nobel Prize in Physiology or Medicine with Philip Allen Sharp for the discovery of introns in eukaryotic DNA and the mechanisms of gene splicing. This discovery led to a fundamental shift in our understanding of genetics. Rich is a native of England who received his bachelor's degree and PhD both in chemistry from the University of Sheffield. Following a postdoctoral appointment at Harvard University, he joined Cold Springs Harbor Laboratory, where he began working with restriction enzymes as a tool for exploring problems of biological interest. This led to the discovery of introns, exons, and splicing. Rich became a consultant to New England Biolabs and chairman of its scientific advisory board in 1974. In 1992, he left Cold Spring Harbor to become the chief scientific officer of New England Biolabs. His current research interests focus on enzyme discovery using bioinformatics combined with the experimental testing of function. He was knighted in 2008 for his services to science. He has received many other honors, including election to the rank of Fellow of the Royal Society and member of the National Academy of Sciences. He is a founder and chairman of the Laureate Science Alliance, a nonprofit organization dedicated to science without borders. His lecture today is titled Nobel Laureate Campaign Supporting Genetically Modified Organisms. Please join me in welcoming Sir Richard Roberts.
Well, it's a great pleasure to be here. It's been a long time, since May of 1969 was the last time that I visited Oak Ridge. It's changed just a little bit. What I want to do today is to talk about something not that I work on, but something that I've become very interested in and very passionate about for a series of humanitarian reasons, excuse me, more than anything else. So one of the things that I've discovered after I won the Nobel Prize was, whereas before I would talk to people and sometimes they would politely listen and sometimes they wouldn't, but after I won the prize, if I talked, people tended to listen. I don't know why, but anyway, the bottom line is that as a result of that, I decided I would try and organize the Nobel Laureates to do some good things for humanity when it was possible. And we've done a number of things. I've been quite successful at getting the laureates to back me. I've generally picked topics that they're very much in favor of. And GMOs and support for GMOs is one of them. And this is a really crucial issue, not here in the US or in Europe or in Japan or in the developed countries, but for the developing world, this is a crucial issue. And there is so much bad science that is being propagated by the anti-GMO people that we have to find a way to stop it. And I always very much disapprove of the misuse of science, and this is one such issue. I'll give you a little history about how I got involved in this for reasons that are interesting too. And then I'll tell you just what GMO plants are, what it means. I know there are people here who study things like physics and don't necessarily fully appreciate what GMOs really are. And an awful lot of the people who are against GMOs have no idea what they are. And there was a very good show that was put on by Jimmy Fallon, who went out and was talking to people and just picking them off the street and say, you know, would you eat GMOs? Would you buy GMOs? Oh, no. What are GMOs? Oh, well, I don't actually know, but I know they're bad for me. And I think this is a problem. We have not done a very good job, and we do not do a very good job as scientists of educating the general public. We don't go out and talk enough to people in the general public, and I think we should. I always give my students something I call the grandmother test. Whenever they're doing something interesting in the lab, I say, go, talk, and show your grandmother, teach your grandmother, tell her what you do, so that she can be very proud of what you're doing. She can actually understand what you're doing. But the key test, the grandmother must then be able to go and explain it properly to her friends. Then you know you've done a good job of teaching. And I think this is something that every scientist should do, maybe not just to your grandmother, but to the general public, to local schools. I go out to local schools and talk. And it's a wonderful experience because very often these young kids, they ask the darndest questions that I don't know the answer to. And for a scientist, this is the very best. This means there's probably something to be discovered there. So let's get on with GMOs. I got involved in this because I was invited to the 80th birthday symposium of the gentleman at the top right, Mark van Montague. And he was the person who discovered the way in which a bacteria, Agrobacteria tumefaciens, was able to transfer DNA from a plasmid that it had in its cell into the plants that it would infect. And in this way, it was able to form crown gall tumors. And we now know there are many bacteria that have specialized in transferring their genes between kingdoms over to plants. At the top left, the person with the beard is Jeff Shell. He worked closely with Mark van Montague during this discovery. And at the bottom is Mary Dale Chilton, who independently found the same method, and they discovered this thing called TI plasmid transformation. And this is just a way of transferring DNA from a bacterium into a plant, something that the anti-GMO people will tell you is unnatural. Well, if it's unnatural, I don't know why nature is doing it. But what happened at the end of this symposium, I'd been asked to go and talk to the European Commission about the future of health. Now, I don't claim to be an expert in this area, but I thought of a few platitudes I could say and so on. But after listening to what had gone on at this symposium, I discovered that after a day of listening to plant biologists talk, and mainly plant biologists in Europe, they were having a real hard time of it. They would go out and try to talk in public, and Greenpeace and the anti-GMO people would come and harass them. They'd cause them problems. They'd try to get them fired from their universities. Just terrible what was going on. And I thought that this was really something that one should try to do something about. Now, the anti-GMO people always attack the plant biologists because usually they get funding from industry. And so they say, oh, well, you can't believe anything these people say because they're just shills of industry. They're just there trying to advertise industry. Now, it occurred to me that of the Nobel Laureates that I know, there is not one of them who is a plant biologist. And I thought, maybe, just maybe, we could do something. So when I went to the European Commission to give my talk, I used this as the title, food is medicine. For the 800 million people who go to bed hungry every night, they're not interested in the fancy drugs that these pharmaceutical companies make. They just want food. And for them, food provides not just the nutrition they need, but also the vitamins and everything else that they need. And I gave this talk about 15, 20 minutes to the European Commission, and afterwards, an Italian senator, a lady from Italy, came over to me, and she said, before I heard you talk, I'd been violently opposed to GMOs. I thought these were dangerous, I'd been talking out against them, and I've changed my mind completely. And she said, just a short talk from me, she had changed her mind. And a number of the staff members of some of the other people, some of the other senators came over and said, you know, this was great. We don't hear this sort of talk in Europe very often. And because the people who talk about it are the plant scientists, and they get harassed, and most of them have stopped doing it. They won't go out and talk to the general public because it's so painful. And that gave me the idea that maybe I could do something. So in December of 2013, I got the idea of running a pro-GMO campaign. And I talked to a few of my good Nobel Laureate friends who were good biological scientists and said, you know, how do you feel about this? What do you think about it? And they all said the same thing. GMOs, there's no reason to think they're dangerous. And all of this talk that we hear from the anti-GMO movement is just anti-science talk. It's just a way of trying to scare people. You know, the oldest political trick in the book is you scare people and then tell them how you're going to save them from this thing you've just scared them about. So I planned this during 2014, 2015, talked to a lot of Nobel laureates, gathered a number of signatures, and we had a formal organization followed by a launch in June of 2016. Ran a press conference down in Washington, got lots of coverage. There was an amazing amount of news coverage. And we basically called on the Green parties to just stop scaring the public by pretending that GMO foods were dangerous. You know, if you don't want to eat GMO foods, don't eat them. You know, you can go to the supermarket, you can buy whatever you want. You don't have to buy organic or non-organic, or you have so much choice. But if you go to the developing world, they don't have the choice. They need better plants, better crops, things that produce more nutrition, and they don't have access to the really good crops that have been bred in the West. You know, one of the reasons that we have so much food here in the US is because the agricultural scientists, both in the companies and in universities, have been improving the crops so that now you get nice, good yields of the things that you want. And in this way, we're able to provide huge amounts of food, a great deal of which gets wasted, unfortunately, in this country. But in the developing world, no one has spent the time to try to improve the crops. There's not been the money. Doing it by the traditional methods that the plant scientists and the agricultural people have used is very time-consuming. It takes 30, 40 years sometime to develop a new crop that is better than something we've got already. And so we really just asked the Green parties to stop scaring people and admit that this was an issue they simply got wrong. You know, when they first came out, when Monsanto first introduced these GMO crops into Europe, there was a huge outcry. And I'll tell you more about the background behind this in a moment. But before I get there, I just want to go and make sure that you understand how GMO crops are made, how they differ from traditionally bred crops. This was the poster that we used. The cartoon up on the top right was produced by a cartoonist in Iowa, published in a paper there. And on the bottom right is a website, supportprecisionagriculture.org, which documents the Nobel campaign and has a lot of useful information connected to this, and it will have my slides on it by the day after tomorrow. You're free to download anything you might want. Now, of course, food means agriculture. You know, when the hunter-gatherers were going out and getting food every day, and then they discovered they could plant some of these plants in their backyard and grow it, they were only growing for their family, much as most smallholder farmers in the developing countries. But if you want to feed a population in a city, they can't grow their own food. They need a good source of food. They need agriculture. And so for some 10, 12,000 years, we've been using pretty crude genetics in order to do this. But in the 1980s, thanks to Mark Van Montague, Jeff Shell, and Mary Dale Shilton, we learned how to do this precisely. Now, this is really a bit of a tongue-in-cheek slide, showing you how you can tell when you've got a genetically modified crop. The non-GMO version of corn, the original teosinte, which was a wild ancestor growing in Central America and South America, was this really thin, spindly thing, didn't have nice grains. And the civilizations in Central America figured out how to select better and better versions of it. They learned how to cross plants. And eventually, we got to the nice corn cobs that you buy today, and which look nothing like the original teosinte. Now, these were all bred, all genetically modified, using traditional breeding approaches. But you could, in fact, do it much better. Now, this is what conventional breeding looks like. We basically find some variety that we're growing. I've got a rice that I'm growing, and I want to make it grow a little taller, or I want it to produce better grain, just to do something better. And so I find a wild variety of rice that can do it better, but maybe it has other traits that we don't want. You can make a cross between these two, and then select from among the hybrids the one that has picked up the trait that you want. Now, when you do that, of course, you end up with this first hybrid, which really is a hybrid. It's got half its genes from one parent, half from the other. Just as when we make human babies, the babies don't exactly look like the mother or the father. They picked up some traits from both parents. Now, that's you really don't want, because many of these genes, the yellow ones here, are probably not the ones that you would like to have. And so what you do is you now make a cross between this one and the original elite variety. And in this way, half of these genes get mixed in here, and you slowly reduce the yellow genes. You slowly get rid of the ones that you didn't want, and you hope to end up with the final variety that has the gene you do want, but it always contains a few that you don't want. But hopefully you've selected something where that doesn't cause a problem. So that is the conventional breeding approach. Now, if that doesn't work, we have another approach that the plant breeders use called mutagenesis. And so they will radiate the plants, or they'll treat them with chemicals to produce mutants, in the hope that in this way they can actually produce the hybrid, the new plant that they want. Precision breeding, the GMO method, this is the one that Van Montague and co. discovered. Here, you take the plasmid that contains the gene you want. So you isolate the gene that you want, put it into this plasmid that Agrobacterium has, and in this way, you can cross this gene into the plant. And so this is a precise way of taking a known gene and putting it into a plant. Now, it doesn't always go exactly where you wanted it to go, but usually you can select a version where it has gone where you wanted it to go. And with new techniques like CRISPR, you can do this very precisely now. It is really easy to move a single gene into a plant. On the left here, you will see the conventional breeding I just described, which Greenpeace says this is very safe. We've been doing it for years. It must be okay. On the right is precision breeding, which Greenpeace says this is dangerous because the gene came from somewhere else. It might even have come from a different version of the original plant. So you could sometimes just isolate this gene and put it in. So in this case, it's doing exactly what conventional breeding is doing. Nevertheless, this is dangerous. And so what I try to do is to give you a concrete example of what precision breeding, GMO, is compared to conventional methods. So I have two cars. I want to move the GPS from one car that has it into the other one that doesn't. Do I take the two cars apart, mix everything, and then select the version that has picked up the GPS system? Well, no, actually, we don't do that. We're a little more sensible. We tend to unplug the GPS from one car and plug it into the other one. What if I take the GPS from an airplane? Does this mean that the car is now going to fly because the GPS came from an airplane? Talk to Greenpeace. They will tell you this is inherently very dangerous. Maybe if you take it from a fish, the car's going to swim. It's just unbelievable, the stories that they tell. And you can imagine to an uneducated public, which most of the public is, they don't understand the science here, it becomes very scary. And in this way, they were able to scare everybody. And really, what is important here is the product. It's not the method by which you make the product. You know, you take your wife out or you take your husband out to buy some new piece of clothing. You don't immediately go and ask, how was that clothing made? You look at it, you say, hey, this fits nicely, it looks good, I look good in it, and you buy it. It's the final product that is important. And if you're going to be testing something, you should test the final product, not the way in which it was made. And unfortunately, the regulatory system in Europe, and to some extent here also, pays attention to how something was made if it's a plant. Don't worry so much about the method by which it was made. Now, one thing that's worth noting is that plants have a big problem. When someone comes along to try and eat them, they can't run away. If a lion comes and tries to eat me, I scoot into a building or up a tree, I get away from it. Plants can't do that. And so you have to ask, well, why are there still plants on the face of the earth? How is it possible that the predators haven't just eaten them all? Well, the answer is they produce pesticides. Every plant you eat has natural pesticides in it because this is how it stopped itself from being eaten by an insect. And I like to use the example of celery. I'm sure everyone in this room has eaten celery. Celery is a very interesting vegetable, tastes okay, many ways to prepare it. The ladies who used to cut up celery in order to package it to go for sale in the supermarkets discovered many years ago they were getting a skin dermatitis from the celery juice. And in some cases, they even got skin cancer. And that is because of this compound sorrelin, which is present in celery and in many other plants too. It's a very good pesticide, very good at killing people who want, killing bugs rather, who want to eat, who want to eat the celery. Now, fortunately, the amounts of sorrelin are sufficiently low when you and I consume it, it's not a problem. Our body knows how to deal with it. Our body is very good at dealing with it. If this was a GMO, this would not be allowed for sale. You wouldn't be allowed to sell it because they would say, oh, you're putting sorrelin in here. Sorrelin is very bad. And we would be in trouble. And it just illustrates almost every plant that you eat, all of the main crops produce these pesticides, and in some cases produce rather large amounts of them. So let's go and look now at the sort of politics and the public face of GMOs. Now, here, as I said before, we have very good crops. We don't need better crops. But what happens is that as soon as you go to the developing world, you discover that pretty much all of the crops that they're growing are not that productive. They're not very good because no one's ever tried to breed them to make them better. And the reason they've not done this is because the big agribusiness are not interested, they can't make money at it, and the local farmers are too busy growing as much as they can in order to feed their families, and the scientists have not been able to make much headway here. Now, why would Europe not endorse GM crops so that the developing world could really feed themselves, maybe stop sending immigrants to Europe? Why would they not do this? Well, there's usually two reasons why people do what they do. Number one is usually money. Okay, so when you ask why are people doing this, always look for the money. And then the second thing is politics. Very often it's politically good to do something or the other. So what happened was the Europeans were not very great fans of American agribusiness and Monsanto in particular. The reputation of Monsanto in Europe was terrible, but they had a little bit of a problem. So they can't ban all these big agribusinesses because they're providing all the seeds that the Europeans need in order to eat. Without them, there would be a lot of thin Europeans walking around, which you don't see. So what can you do? What can you do about this? Well, Monsanto made a huge mistake. So they introduced GMO crops into Europe, or they tried to. And they did it in such a way that Monsanto could make a lot of money from it because they charge more for the seeds. The farmers made a little bit of money from it by charging higher prices to the consumers. And so here you've got consumers being asked to pay more for something that was not particularly better, it offered no great value over and above what they'd already been buying at cheaper prices. And this gave Greenpeace the opening. Greenpeace said, we're not going to be against Monsanto per se, we're going to be against GMOs. And so they picked GMO, actually a man called Patrick Moore, who was head of Greenpeace at the time, came up with this idea of the marketing strategy. So the idea is you assert that GMOs, precision agriculture, is dangerous. You produce wonderful scenarios whereby you scare people. Very easy to scare people. You know, I'm sure you all like going to horror movies. Really, they're very good entertainment, and the horror can be particularly horrific. At the end of it, you sort of know that it wasn't real because you know it was just a Hollywood movie. But nevertheless, there's this little bit of remnant of fear left in your head. Well, Greenpeace made sure that that fear was well and truly planted in everybody. And so they talked about all of the horrible things that might happen. And what they did at the time was to say, well, one of the things we need to know, before we start planting these things all over the place, we've got to know if they really are dangerous. Are they going to take over the world? Are they going to prevent other plants from growing and so on? And they call for experiments. Now, Greenpeace claims to be an organization that follows the science. The science has now been done. The experiments showing these things were safe were done many years ago. Why hasn't Greenpeace changed its mind? Why hasn't it just said, this was an issue, we got it wrong. We were right to call for experiments, we were right to call for testing because we didn't know what was going to happen. Now the experiments have been done, there's been absolutely zero danger shown. Why don't we just admit we were wrong and get on and do something else? And the answer is because this has been the best fundraising move that Greenpeace ever had. They're an NGO. Their annual budget in Europe is estimated at about 400 million euros. This is how much money they get in, and most of that comes because of their anti-GMO stance. And the nice thing about this entire campaign was it had no consequence for Europe. Europe didn't need GMOs, and the people were happy to be paying the lower prices. They didn't want to pay higher prices for GMO products. But the problem is that as soon as you start to say these things are dangerous, you cannot just say, well, they're dangerous for Europeans, but they're perfectly okay in Africa. And so they now go around talking to everybody that will listen to them around the world, claiming that these things are dangerous. It's really bad. Now, the scientists have tried to respond to this to some extent. Every major scientific society in the world, National Academy of Sciences, the Royal Society, national academies everywhere have all come out and said there is no reason to think that GMOs would be any more dangerous than traditionally bred plants. There are only two supposedly professional organizations that have opposed it, and these are two organizations that were put together because of their anti-GMO stance, and so they just put out anti-GMO literature, anti-GMO sentiments whenever they can. So something like 230, 240 academic societies have all said they're safe. Anti-GMOs said they're not. Why doesn't Greenpeace listen? Why don't the anti-GMO people listen? Now, what I would like to do is to give you some examples of why the developing world really needs GMO plants. And the first point I would like to go at is to look at this problem of vitamin A deficiency. If you do not get enough vitamin A during the first few years of life, you stand a chance of going blind. It's very important for the development of the retina. You also have problems with autoimmune diseases and with development in general. And for a very large number of people, a large number of young people, they have vitamin A deficiency. So you can see here somewhere like 2 million children die under the age of 5, die every year because they didn't get enough vitamin A. For a lot of these people, the problem is rice is a major portion of their diet, and rice does not contain beta-carotene in the grain, which is the precursor of vitamin A. It has it in the roots and in the shoot and in the leaves, but it just doesn't put it in the grain. And so two scientists, Ingo Petrikas and Peter Bayer, two European scientists, decided maybe there's something they could do about this. Maybe they could convince the rice plant to put the beta-carotene into the grain and then provide vitamin A. And they tried to do that by traditional methods and couldn't do it. And so they chose a GMO method to do it, and they were successful quite quickly. And the bottom line was that in 1999, golden rice, the very first golden rice, it's called golden rice because beta-carotene is yellowish in color, and so the golden rice has this yellow character associated with it. Now, typically, when you make a product like this, you make a plant like this, it takes 2, 3, 4 years to get it growing out in the fields and to show that it's robust and will work. But it's a GMO, and as a result of that, it's not going to be available until later this year. You know, this is 20 years since this was first developed, and only now does it look as though it may be a product. Why? Well, because the anti-GMO movement saw that this was a potential pharmaceutical product, and they were very worried about that. You know, I'm sure there are probably some people here who have diabetes, and if you're diabetic, you take human insulin. And you know where that human insulin comes from? It doesn't come from humans, it comes from a GMO. It's a yeast that has had the human insulin gene put in it, and it makes insulin, and we all take it, and it's perfectly good. Have you ever heard the anti-GMO people complain about that? They don't tell the diabetics, oh, this is going to kill you because it's a GMO product. No, it's a pharmaceutical, and they know better than to try to complain. And golden rice, they saw that this was a potential problem for them. And so what they did is they put every conceivable roadblock in the way of its development that they could. And then they had the nerve to produce this document called Golden Illusion. They said, rice, golden rice, it's been so many years since it was first thought out. It's not developed, no one's using it, it can't go, it can't work. You know, it must be wrong. There must be something wrong with it. They didn't mention the fact that it was they who had stopped this development because that didn't fit with their story. And the bottom line is that since 2002, more than 15 million children have died or suffered because of vitamin A deficiency. You know, if this was a problem in Rwanda of the young children dying because of actions by the Tutsis, it would be in everybody's sight. The UN would be all over it, the diplomats would be, everybody would be against it. But in fact, because this is just a product of the action of the anti-GMO movement, nothing's happened. Nobody gets worried about it. I want to know how many kids have to die unnecessarily before we consider this a crime against humanity. As far as I'm concerned, this is a crime against humanity. It's not right. We can do something better. Here's another interesting case, bananas. I'm sure you all like bananas. Bananas are the perfect food. You know, they come pre-packaged. You take the packaging off, and then you eat it. No problems. However, bananas do have a problem when you're trying to grow them. So bananas have been so heavily modified over the years that the original bananas were pretty much all seeds. You eat one today, there's almost no seed. It's actually quite difficult to find a banana with a seed in it. In Uganda, and much of sub-Saharan Africa, there is a problem. There is a disease of bananas called Xanthomonas wilt. This is a bacterial disease, gets onto the plants, kills the plants, and really devastates them. The only way you can deal with this is basically by spraying the bananas 10, 12 times during the course of their growing period with some pretty nasty pesticides, chemicals, in order to stop it. The farmers who typically grow bananas can't afford that. And so the scientists in Kenya thought there was a solution to this. They discovered that sweet pepper, which is completely resistant to Xanthomonas wilt, had 2 genes in it that were responsible for this resistance. And so the scientists thought, let's take those 2 genes, put them into bananas, and maybe the bananas will be resistant. And the bottom line was, yes, they were. They grow happily. They're very nice. And the thing that is problematical here is that there is no other route to get to these bananas that are resistant to Xanthomonas wilt. There are no natural varieties of bananas. Some 400 or 500 have been tested in Belgium. There are no natural bananas. So you can't do it by traditional breeding. This is the only way that you can make these bananas resistant to Xanthomonas wilt. Why would you not want to do this? 30% of the calories consumed in Uganda all come from bananas. There was a bill that went through the parliament in Uganda to allow the production of GM bananas. And it was overly regulatory, but nevertheless, it was sufficient that it would have allowed the farmers to grow it. It went to the President Museveni. At first, he is very pro-GMO, I should say. At first, it looked as though he was going to sign it. And then we heard that his wife and his daughter had gotten to him. His daughter is very religious. She'd been gotten to by some pastor in Canada who had been telling her how dangerous GMOs were. She then convinced her mother, and the 2 of them ganged up on the president and persuaded him to send the bill back to Parliament with a whole bunch of strict restrictions put into it that effectively made it worthless. No one would do it. It was not possible to do it. And it's now sitting with the Parliament, and they're trying to figure out what to do with it. And yet these things grow, and the people need them, and the banana plants are dying. If this isn't solved, then the only bananas people will eat will be ones that have been heavily treated with pesticides so that this Xanthomonas wilt doesn't cause a problem. Fall armyworm is another big problem. This used to destroy the corn crop in the southern part of the US until the scientists came up with a way to stop it. There is a bacillus called Bacillus thuringiensis that produces a toxin, Bt toxin, that will kill the fall armyworm. It kills a lot of insects, actually. This is the pesticide that the organic farmers use in order to grow their crops and keep the crops down. So organic, it's organic. The organic farmers think it's okay. They spray it. However, they said, if you take this gene and put it into corn, it's a GMO, it's dangerous, must be labeled, no one should do it. Well, in fact, fortunately, this was done before the anti-GMO movement really got going, and all of the corn now grown in the southern part of the US is Bt corn. The South Africans had this problem with fall armyworm. They knew about the Bt corn, they shipped it over to South Africa, it grows perfectly well there. But you go to Zimbabwe, you go to Malawi, you go to some of these other countries, you discover that the presidents of these companies have instituted a complete ban on GMOs because the Greenpeace allies got to them, convinced them they were dangerous, and so they put a ban on. You can't grow it, no growing. And so the farmers there are now looking at their corn crops and seeing that the yields are dropping. This disease spreads incredibly rapidly. It's spreading throughout southern Africa where they're not growing Bt cotton, and it's causing problems. People are going hungry because of this when there's a simple solution. And this has been grown in this country for years. GMO corn is eaten by billions of animals. It's even eaten by people. Have you heard of any problems with it? No, there have been no problems with it. It's not a problem. It's just a very straightforward solution to an awkward problem. Now in Bangladesh, they have been better than almost any country you can think of. You know, in Bangladesh, they face major problems because of global warming. They're going to lose about a quarter of their land as sea levels rise. They have a huge population. There are about 180 million Bangladeshis, and they have problems feeding them. One of the staple crops there was brinjal, this eggplant, BT eggplant. And so they were having problems with diseases. Someone made a BT eggplant. They started growing those. They grew very successfully in Bangladesh. Laws were put in place to allow the farmers to grow them. And last year, they produced more eggplant than they could eat. They've been unbelievably successful in this. Farmers in India, in the neighboring province of Assam, also like eggplants, but in India there's a ban on them. They're not allowed to grow them. And so what do? They go and see their friends in Bangladesh and they smuggle them across and they grow them. Farmers are smart. They're not foolish people. Another interesting case, Hawaiian papaya. So one of the major fruit exports from Hawaii is the papaya. And they've been doing this for many, many years. And then they were hit by a problem, something called ring spot virus. It's a small RNA virus. It's carried by insects, and it was killing the papaya trees. But there was a nice solution. A man called Dennis Gonsalves, a scientist at the University of Hawaii, thought he had a solution to this. And he made GM papaya plants that were resistant to this ring spot virus. Essentially what he did was to vaccinate the plants against the ring spot virus. And now something like 75–80% of all of the papayas coming out of Hawaii are GM papayas because of this. And then the politicians in Hawaii had this great idea. When they heard about the GMO labeling laws that were going on in the US, they decided they were going to have one too. And so they put this up and they proposed they were going to do it. When the farmers heard about this, they were outraged. They kicked up such a fuss and they said, what are you trying to do? You're trying to put us out of business so that we have to label all our papayas as GMOs and no one will buy them? You know, is this good economics for you guys? Well, there were hearings, there were a lot of fuss about this. And so what did Hawaii do? Politicians did. They found a compromise. They said, well, we will grandfather GM papayas. We'll pretend they're not GM, and it's only going to be new things that we make that have to be labeled as GM papayas. In Thailand, they have the same problem. It's a slightly different ring spot virus. The plant that they grow in Hawaii is not resistant to it, but farmers scientists in a neighboring country did exactly the same thing to the Thai papayas, and now they are resistant. But Thailand, anti-GMO. They're not allowed to grow GMO plants. So what do? The farmers just grow them anyway and just don't tell anybody that they're GM plants. Very sensible. So the bottom line here is that if you don't want to eat GMOs for whatever reason, don't buy them. You know, nobody makes you buy GM plants, but it turns out that most people make their food choices on the basis of either taste or price. And for a lot of people, it's price that is key. If you're poor, you don't want to spend extra on a plant that isn't perhaps any better than what you're growing. And if anything, GM plants are probably safer than traditionally bred plants. You remember the slide I showed? These traditionally bred plants always have extra genes in them. What are they doing? We don't know what they're doing. In the case of GM plants, we know exactly what gene has gone in, we know what it does, we know where it's gone, and we can look and see, has it influenced transcription, has it caused problems? And the bottom line is, wherever people have looked, there's been no problems, absolutely no problems. There's not been one accident, one reported problem with a GMO plant, despite now more than 25 years of experience with them, of people and cattle eating them. There's not been a problem. So for us, we don't need GM plants because big agribusiness improved all the plants a long time ago. There's still more they can do. And their method of choice is not to spend 20 years developing a new crop. Their method of choice would be to use the GM method and do it in 2 or 3 years if they're going to make a better crop. Why would you take it slowly when you can do it quickly and, if anything, more safely. But we must never forget that when we make statements in the West and we make political statements, everybody hears them. You can't pretend this is just something for us in the West, because it affects the whole world. And I'm sure we can all agree as scientists that we need more science in politics and ideally a lot less politics in science. And politicians should really listen to the science that they fund. Why do they fund us if they don't want to listen to our results? Climate change is a huge example of this. GMOs is just another issue. Why do they fund us if they don't want to hear what we have to say? They should stop supporting the idea that foods produced by GMO methods are dangerous. They're not. They're simply not. There is no reason to think they're dangerous. If you thought they might be, then you ask that they be tested ahead of time. But if you're going to test GM plants, I say you test all the traditionally bred plants too, because I think the danger there is much greater. And we've had many examples of dangers from traditionally bred plants, like there was a huge disaster with potatoes that had been bred and were producing large amounts of solanine, I think it's called. But anyway, it's a toxin that is not very nice, and they had to withdraw this. There were thousands of acres of potatoes were destroyed that way. And also the EU now are trying to get a passage into their bill, into their constitution, saying that no G8 country should support GMO research in Africa. That I find appalling. I mean, this is just outrageous. But I think civil society can help too. We're in fact trying to persuade the Pope to make a positive statement about this. He believes in the science, he's convinced of the science, but he's been fed some bad information by an Argentinian economist who is a friend of his. And this economist has claimed that the introduction of GMO plants wiped the smallholder farmers out of business in Argentina. It's not true. Big agribusiness was wiping out these small farmers long before GMOs came on the scene. And this economist is simply wrong. I've been trying to meet him and talk to him. He refuses to meet me. He won't talk with me. Greenpeace too, they won't talk to me. Groups such as Rotary clubs can actually be quite powerful. You know, the Rotary clubs are very good. They're worldwide. They care about the poor in their community. And I've given several talks to Rotary clubs, and they're very supportive of this. They like this idea. We would like to get that onto their international agenda. It'd be nice if some influential celebrities should speak out. You have to be a little bit careful because they don't always get the science right, and you've got to pick the right ones. But the media should really make sure that they do a better job. This is not an if this, if that. It's not a yes, no issue. This is an issue where it is clear that most scientists who understand the issues know that GMO plants, if anything, are safer than traditionally bred plants. The media should make this a point. They shouldn't try and present it as a two-sided argument. It really isn't. Now, the most exciting thing I've heard recently that came out of the Vatican, you know, if you're a Catholic and you take Mass, you have to eat some bread and you have to drink some wine. And so they came out with this little doctrine that if the bread is made from gluten-free flour, apparently it doesn't work. However, if it's from a genetically modified organism, it's okay. So this is the best we've gotten out of the Vatican so far. Now, there are some very prominent people who've come up and used to be very anti-GMO and are now totally pro-GMO. Patrick Moore, the guy who started all of this for Greenpeace, has now he left Greenpeace, he read the science, he's flipped completely, he's 100% pro-GMO. Stephen Tinsdale, he was the head of Greenpeace in the UK, he switched. Mark Linus, he's now with the Cornell Science Alliance, and he was one of these people who were going out burning crops in England when they were first trying to do the field tests. He switched completely and is actually now quite an outspoken critic of Greenpeace and the others. But the best news of all is this gentleman at the bottom. He is the president, or was the president, of the Ghanaian Peasants Farmers Society. He was a very big voice anti-GMO because of his position with the farmers. He recently came out about 6 months ago and said GMOs are good. He says, I've read through all the science, I've learned everything I can about this, this is good, and he's now trying to convince the Ghanaian government that they should be pro-GMO, they should take advantage of this. And that's what we really need. We need more leaders in Africa, in India, in China, who will come out and really support this. And my last slide, I just say, have a heart. If you live in the West, your dinner table looks like that on the left-hand side. If you live in Africa, you don't have that same luxury. To be non-GMO is an indulgence of the West. If you don't want it, don't eat it, don't buy it, don't take it. Although it's interesting, there was a recent survey done in Vermont. Vermont was the first state to have GMO labeling laws. And so the survey was looking to see what people were buying. And it turned out whether it was labeled GMO or not made no difference whatsoever. It was price that mattered to the shoppers. So if they want to label it, let them go ahead and do it. This was a move started by the organic farmers. They were hoping that if you had to test to find if you had GMOs in your products, that this would raise the price of the products because you've got to pay for the testing. And then the price differential between organic and regular foods would be less. The organic industry spent more than $50 million on this advertising campaign to get a GMO labeling bill passed. And finally, I would say, please do go and take a look at the website. You will find the names of the 141 laureates who've joined so far. You'll also find names of many people like yourselves who support us. So please, read, look at the site, sign on, and please tell everybody you can think of that GMOs are actually okay. Thank you.
Perhaps we can...
If there are questions.
I'm going to let you sit over there.
Oh,
lead.
Okay.
So I'm going to ask you, you mean to say that I was just sitting and calculating all the money that I spent in buying organic.
Yeah.
You mean to say that it was all a waste of money?
Well, put it this way. If you want to get a bacterially acquired infection, buy organic. Because they have 10 times the rate of bacterial infections. And you can imagine why that is. You grow your crops in shit, well, what are you going to get? So yes, you've wasted your money.
Again, thank you very much, Richard, for pointing that out to me. I just want to let you know that the last both Richard and I go to a conference organized in Kyoto. It's called Lights and Shadows of Technology and Innovation. And it's really a very interesting and thought-provoking meeting. And the way it is organized is that you have, I don't know, a dozen or 24 Nobel Laureates, captains of industry, people from all walks of life, and Koji Omi, who is the former Minister of Energy in Japan, organizes this. It's very well done. This past year, in fact, we had 5 early career staff from the laboratory attend and participate in that. And Richard was very gracious in engaging, and others were very gracious in engaging with the staff. So I invited him to come here and engage with us. And in particular, because one of the things that we are, as a laboratory, we are certainly passionate about is trying to sort of recreate the importance of biomedical research here at the laboratory, even though it is not a mission of DOE. You mentioned that the last time you came was May of 1969.
Right.
You recall why you were here? And
I came to learn some chromatographic techniques.
Okay.
So I was a postdoc at Harvard. I was sequencing a tRNA in the days when that was actually quite difficult. I was told by a postdoc who was already working there, who I was working with, these methods that had been pioneered by Bob Holly in Wisconsin was the way to go, and this was using unlabeled RNAs. And so there were problems in purifying the particular RNA that we wanted. And there was a group here who were very good. They were developing some modern liquid chromatography methods. And so I came here, learned how to do it, went back, the postdoc left. In the meantime, I'd been reading about Fred Sanger and everything he'd been doing. And so I ended up switching, making P32 labeled tRNA, and I ended up sequencing that way. So I wasn't able to take advantage of what I'd learned here. But it was a fun trip. I had my wife, 2 young children, and we spent a couple of days in the Smoky Mountains, being rather horrified at the conditions.
Well, I hope that this visit you will get a chance to pick up a few techniques that maybe we will get a chance to use in your research. Mm-hmm. Going back to the presentation, I think the underlying theme was as scientists, we have an obligation to communicate what we do and the importance of that. And this is something that, as you well know, we all struggle with. And there are a lot of people here who are, we try to do this both as an institution as well as individually. Any particular insights that you would offer to this group?
Well, the one thing, I was very fortunate. So I grew up in England at a time when children who were in school had to make presentations regularly to their classmates about something they'd read or some homework they'd done or something like that. And if you didn't do it very well, the teacher would point out, well, you should have said this and you could do this. And so we were really taught how to speak to people who were not experts in whatever we'd been reading. And I found that really, really good, really helpful. And I think we should make sure that all young scientists in particular, but ideally all school children, really learn how to do this. This should be an essential part of education. Because you know as well as I do, there are times in your life when your future depends upon how well you present yourself. You go for an interview, you should learn how to do this as well as you possibly can. And it's never too late to start learning. There are actually some quite good online courses now that are being taught that can help people. But the bottom line, practice is what really works. So find an audience and practice on them. It can be your family. You can bore them to tears while you practice.
I was struck, we were having this sidebar conversation before your lecture, and one of you have not had a chance to visit, the full agenda today, but your sort of instinctive reaction was that we are too spread apart, mm-hmm. and that the kind of science and research requires greater interaction, greater multidisciplinary engagement. To some extent, that is something that the labs, national laboratories, pride themselves. So, obviously, science is driving us there. Are there any particular, advice or thoughts that you have in terms of how one could encourage greater engagement?
Well, I think you need to find out what people are doing in other buildings. And so you need to make sure there are opportunities where you can interact with people in other buildings and be sure that you're aware of what they're doing. And ultimately, many good collaborations just end from good personal friendships. And so, if you find someone working in a different area and you become friends and you start to talk about what you're doing, very often you discover, oh, I could use this technique in order to do this. Or you can talk to the other person and say, well, I have this problem, how would I solve it? And maybe they can't solve it for you, but maybe they have a friend in one of the other buildings who they know, oh, this person's interested in doing. So really, there's nothing better than talking to other scientists. And the problem is when the labs become separated, you tend just to talk to those people who are in your local vicinity, and you don't get exposed to the full range of things.
Well, there are people with microphones here, so I think I give the audience an opportunity to ask questions of Richard. Yeah.
Just speak up.
Sure.
Okay.
Yeah.
Yeah, microphones. Hi. Thank you very much. When I speak out in the public about biotechnology, I often bring up at least the golden rice and some of the other examples. So I actually have a favor request on your site. Could some of your slides be made available, or are they already up on your site?
Yes, they're already up.
Great.
Maybe they're not as easily found as they could be. I perhaps can fix that. But yes, they're all up on the site.
Okay, because
you're welcome to use them.
Because sometimes that's, well, you know, we're
Yes.
busy.
Right.
This is not in our primary area.
Right.
And when we have to talk broader questions, that's sometimes a challenge.
Yeah.
Thank you.
Yeah, you're welcome.
Can you introduce yourself, please?
My name's Tiago, and I'm with the Iron Source guys here at SNS. So most of the criticisms that I hear about GMO are actually about business practices. So could you comment more generally on the role of business in making GMOs more palatable?
Yeah, so, this was Greenpeace's grand strategy. They took advantage of the fact that the Europeans hated big business. They hated the agribusiness, and they just welded the GMO issue into it. But they're two totally separate issues. So I can certainly talk about agribusiness practices. I think they're just like the pharmaceutical industry. They're like any big business, any big capitalist business. They do what they do to make money. They're not interested in being particularly philanthropic and helping the public. They're in it to make money, and they do everything they can to make money. And I think they have done the world a great disservice by introducing things like GMO crops and then expecting farmers and consumers to pay more for them when there is no value, no added value. And this was Monsanto's big mistake as far as I'm concerned. Had they introduced a trait that really would have, made things taste better, maybe people would pay a little more for that. I'm sure you're all aware that most of the fruit that you buy in this country doesn't taste quite as good as it used to. And this is because the traditional plant breeders have been looking to produce more and more and more apples on a tree rather than make apples that taste better. And so I think this is where agribusiness has failed. They've been really focused on making mass amounts of food available at prices that people are prepared to pay and where they can make as much money as possible. And I think the pharmaceutical companies have exactly the same problem. In fact, most big businesses, this is the problem. They're in it to make money. It's the downside of the capitalist system, that they don't really think about the people who are poor, who really need these products and can't afford to pay very high prices for them. That's not what they're thinking about. And this is why they've done nothing with the crops that grow in the developing countries, because they can't make money there. People don't have the money to spend.
So if I may add, do you see business as part of your campaign?
Sorry?
Do you see getting business on board as part of your campaign to...
Not really. I mean, I don't want to take on capitalism. I think some economist needs to do that. No, what I do try to do is to separate the two issues, to make sure that people are aware that we're talking about the GMO technology and what it can produce, not how it can make more money for business.
Thank you very much.
And on that note, the nice thing is that while certain things, all agricultural products have patents on them, many of the improved crops that is needed in the developing world can be made by scientists and companies in the developing world. And so they can also use this as a way to improve the economy, the economic health of people there too. So for instance, BT cotton, which is now grown in India, has been incredibly successful. And the farmers there are very happy that they're growing it. And the BT cotton, actually, a lot of it is sent to Europe, where they manufacture the euro notes from it. And so I always tell the Europeans, if you don't like GMOs, give me your money.
This right there.
Yes.
Further back.
No, it's coming.
Hi, I'm Alyssa Carroll. I'm with the Plant Bio Group. So here you presented like 2 organizations that are against GMOs, and they're getting like massively broadcast across over like 200 organizations that are saying it's safe. And I think this happens a lot in other sciences too, like in global climate change, that they kind of get drowned out by these few people that are against it. And some of your advice is that we need to obviously present our science, but the rest of us we're not Nobel Laureates. So, and you mentioned like celebrities, like should we start writing campaigns to Britney Spears to promote the true science? Like how do we get our message out to the public when we don't have the same platform that you do?
Can I answer that?
Sure.
You first get your Nobel Prize.
Oh, okay.
Yeah. Well, I think, it's always nice when you can reach a big audience, but I spoke at a local school just a couple of weeks ago, and I spoke to 11 and 12-year-olds, and one of the kids wrote to me afterwards and said that they'd gone home and she had talked to her parents who had didn't like GMOs, they were opposed to GMOs. She said after she talked to them, on the basis of what I had said, they had changed their minds, and now they were quite happy with GMOs. They weren't worried about it anymore. So teaching kids, talking to kids, getting them on board, this is a great way to do things because they go home and educate their parents. And kids on the whole are much more open to new ideas. They don't already have these ideas fixed in their heads, unless maybe, they come from a religious family or something. But in general, kids are much more open to new ideas, and they're very open to logic. They understand logic. So you don't have to have a massive audience to have quite a decent effect. But that's why we all need to do it. I can't talk to everybody. No one can talk to everybody. Maybe the Pope can talk to a large number, but that's what we're hoping to persuade him to do.
Well, in the interest of time, I'm going to try to close this up. But since we have you here, I personally believe that sort of modern biology today is where information technology was 25 years ago. So, just sort of stepping back in the field of biology, or from your vantage point of view, what are the 2 or 3 really exciting scientific, directions that you see as emerging that we all should be paying attention to?
Well, the thing that I'm most excited about at the moment is the microbiome and the role that bacteria play in our lives. If you just look at it from a human standpoint, if I give you an antibiotic that kills all of the bacteria that are associated with you, you will die. You can't live without them. And a good way to think about this is that bacteria have made us their home. And what you do when you've got a home, you put a fence around it, you put an alarm system in. If you live in Texas, you buy a gun. But bacteria do the same. They don't want anything to happen to us. And so 99.9% of all the bacteria living with us, they're trying to protect us. They don't want us to get ill. They found ways of combating pathogens and stopping pathogens from growing, and we don't understand how they do this. We also know they produce anti-cancer compounds. They don't want us to get cancer. How do they do this? What are they doing? And the most nerve connections between the brain and any other organ in the body is the gut. There are more nerve connections leading from the brain to the gut. And it's clear these bacteria are talking to us. I want to know what they're saying. So that's an area, but this applies. Every single living creature has a microbiome. And in general, the microbiome is an essential part of its life. And we understand very little about this. Soil is one massive microbiome. I'm sure there are ways in which we can remediate soil if we understand the microbiology of what is going on in the ground. And so this, I think, is something that's very exciting. I also think there are lots of excitements in sequences, in analyzing sequences, figuring out what it all does, ultimately we're going to know everything about the organisms on this planet because we will have their DNA sequences. The challenge is not to get the sequence anymore. The challenge is to understand how we interpret that sequence. How does a sequence make an organism work? And I'm particularly interested in bacteria because they're nice and simple. I find humans way too complicated, but bacteria are great. I love them. I am hoping that before I die, we will understand how a bacterium works. That means we will know enough about the signals that are being produced, the proteins being produced, the metabolism that's going on, so that we could emulate it in a computer and program a computer that will behave exactly like a bacterium, and then we'll know that we really understand it. That, I think, would be very exciting, and it's going to lead to a lot of new things being found that affect the biology of all of us, not just the microbes. So that I'm very excited about computational possibilities, but it's not just about sequencing, it's about trying to understand what the genes are doing, when they're transcribed, when they're made, and so on. So that's a couple of things for you.
Thank you so much. So please join me in thanking Sir.
Thank you.